Hello everyone. So today we are going to discuss the most important topic of the NCRT book of chapter biotechnology of class 12th, that is PCR. PCR stands for polymerase chain reaction. As poly means number, many number of things. So here it stands for number of DNA copies, number of recombinant DNA, and in the chain means that is getting repeated once it ends and again it get repeated, and reaction as it will the process, so it will be a reaction. So it is given by the Carl Mullius in 1983. So for amplification of gene we use PCR, which also mean formation of many recombinant DNA. In this reaction, multiple copies of a gene or a DNA of interest is synthesized in vitro using two sets of primers. Every strand has 3 dash 5 dash and okay, and I'm also going to teach you by making a diagram, theoretically also and practically also. Okay, so PCR is done in the three steps or three process, we can say. These three steps are as follows. Number one is denaturation, or we can also say a denature. It's up to you. And the second is annealing, or you can also say anneal. uh, and the third one is extension, or there are a number of names which are given, but according to NCRT it is extension only. So now we are going on the denaturation. Denaturation is done under the temperature of 90 to 95 degrees Celsius. In this, both the DNA are get separated at at the temperature it is given. The temperature is depend up to the place, and it's get vary up to place. Now in annealing means to anneal. It is done under 40 to 60 degrees Celsius. Primers are formed at 3 days. We use MG2 plus 2 here. It's up to the place where we are going to do the process, because it's not a small process. Number of particle, number of DNA technologies are used beyond it. So it is done at the 3 dash, and now we are going on the extension process. So at the extension, as extension means to increase extent. Okay, so the new DNA form at the both side. It this process and under the temperature of 72 degrees Celsius, and the temperature may vary here too according to the place where you are doing this process. Okay, so this all I have explained you. This all are the process by which we can make the new DNA. Okay, now I am going to tell you by the diagram, like how the process is being done, actually how it is done. With me, you can also try in your rough notebook, so it can be a practice as well as you can learn some new topic if you are not knowing this. So here I began. So these are two primers which I have marked. You can recognize it. So the small chemical is synthesized oligonucleotide that are complementary to the regions of DNA. This set of primers are too beneficial for the next process of the extension. For this, we are using DNA polymerase, that is the enzyme which we use if the temperature may not be very. So here I am marking also the process, like first one is denaturation, second one is annealing. So you can understand easily, and you have to draw like this only in the board. Surely you will get the full marks. Now we are moving forward. We are using here the Taq polymerase. We are also using a deoxynucleotide. You can write that too also. For moving forward with the next process, that is extension, which is done under the 72 degrees Celsius. So it's compulsory to mark this 5 dash 3 dash. So here 30 cycles has been done and number of copies have been made. The whole purple part is amplified DNA copy. This was all about the PCR. If any issue, so you can DM me in the comment. Okay, thank you.